Hello everyone, I'm Abir Al-Mazloum and today I will present a project under the title of Targeting Cardiomyocyte Survival for Cardioprotection Against Chemotherapy. Cancer is one of the leading causes of death worldwide, and chemotherapy is the most effective treatment. One of the most used chemotherapeutic drugs is doxorubicin, which was shown to be effective against different types of cancers. However, despite its effectiveness against cancer cells, doxorubicin also damages other cell types, including heart cells. And when the heart muscle is damaged, it fails to supply the organs with enough blood, which can cause organ failure. And in the short videos below, we see the difference between the normal beating heart and a weak, stiff, failing heart. So patients who are surviving from cancer are having heart problems later, which can be fatal. And when we look closer to the heart, we notice an increase in scar tissue, which is causing the stiffness. Normally, cardiomyocytes, which are the beating unit of the heart, are surrounded with many cell types, including fibroblasts. But when under stress, such as treating with doxorubicin, cardiomyocytes die, and those cells do not regenerate, so they are replaced by fibroblasts, which produce the scar tissue. Therefore, we collaborated with a medicinal chemistry lab to synthesize a new molecule that we called LCB2122. And in vivo experiments were done on mice where they were treated with doxorubicin after surgically implanting mini pumps that deliver a specific dose of LCB2122 per day. Echocardiography analyses are done and show that in mice treated with doxorubicin, the ejection fraction, which is an indication of the cardiac function, representing the amount of blood pumped in one beat, is significantly decreased, while when treated with 2122, the percent of ejection fraction is higher. Hearts are also taken to assess cell death, and as represented in the graph, LCB2122 was able to decrease doxorubicin-induced cell death. Now we want to know how this molecule is protecting against cell death. And since the structure is similar to an AMP, we hypothesize that it's activating the AMPK survival pathway. AMPK is one of the most important energy sensors in the cell, which is activated by a low ATP and high AMP levels. The AMP binds to the gamma subunit, causing the phosphorylation and thus activating AMPK to regulate several pathways, including mitochondrial functions and lipid metabolism, by phosphorylating and blocking the acetyl-CoA carboxylase. Now, in neonatal rat cardiomyocytes, after 6 hours of treatment, doxorubicin increases cell death more than twofold, while LCB2122 was able to protect against doxorubicin-induced cell death. Now, upstream to this AMPK, the kinase LKB1 is responsible for AMPK phosphorylation. So I treated neonatal rat cardiomyocytes with LCB2122 and blotted for the phosphorylation of AMPK, which is significantly increased after 1 hour, in addition to the phosphorylation of LKB1, which is also significantly increased, and the downstream acetyl-CoA carboxylase phosphorylation is increased after 2 hours of treatment. Now, another very important post-translational modification of LKB, which is known in the heart, is the acetylation. This acetylation makes LKB inactive. So in order to be active, sirtuins, which are the deacetylases, remove this acetyl group and making LKB active. To check the activity of sirtuins in presence of LKB2122, I did an in vitro assay which shows that in presence of 2122, the SIRT1 activity is significantly increased as compared to the controls. Now, to make sure that this protective effect is through AMPK pathway, we deleted AMPK by using siRNA In the control groups, doxorubicin increases cell death, which is significantly decreased when treated with LCB2122, even more than the known AMPK activator ACAR. However, both 2122 and ACAR were unable to protect against doxorubicin-induced cell death in AMPK-depleted cells. Now that we know the mechanism of activation of LCB2122, our next goal is to move to bigger animals as a preparation for clinical trials.